Homologous recombination. Now, the word recombination is basically suggesting that an existing combination can be changed to get a new combination. And interestingly, this is what is happening in our chromosomes and DNA, where gene rearrangements are possible, and these gene rearrangements enable variations that are useful to the cell, or variations that can help the cells evolve to become better. So let us look at the learning outcomes of this session. So homologous recombination is a phenomenon that is prevalent in cells to enable rearranging, exchanging of DNA between chromosomes, especially if it is a homologous pair of chromosomes. Being an essential cellular process, the entire mechanism of homologous recombination is aided and regulated by different proteins and enzymes. This process enables genetic variations due to exchange of genes, retrieving sequences to repair damaged DNA, and in certain cases, it can also regulate gene expression. Due to its specific mechanistic, it is being used also to modify genes in recombinant DNA technology to create knockout organisms and transgenics. In this session, we are basically going to focus on the model of the homologous recombination, the mechanistic, which is observed with many models that are present for homologous recombination. So the model that we are going to discuss, or that we are going to look at in this session, is what is called as Holliday model. And Holliday model, in fact, is due to the scientist Holliday, who proposed how the crossing over, or how the exchange of genes takes place. And the typical structure that is formed because of exchange of genes is what is termed as the Holliday Junction. Now, breaking and joining is an inherent part of recombination mechanistic. This is something that has been found in any model that has been used to explain how homologous recombination occurs. Now, what will be done is that there are certain key steps, there are certain basic steps involved, whether you take the Holliday model or whether you take the double strand break repair model, any of these, you have a certain number of basic steps, and that is what we are going to go through. So step one is you have the aligning of two homologous DNA happening. So as you can see, you have this DNA, which has A, B, C as dominant alleles of these genes. And this is a homologous pair, homologous dominant as we say. While this one is identical or closely identical to this one because it has the same genes, the only difference being that the alleles are recessive alleles. So this is a DNA which has dominant alleles, and this is a DNA which has the recessive alleles. But as you can very clearly understand, the genes are the same. So this is gene A, this is gene A, this is gene B, this is gene B, this is gene C, this is gene C. So the positions also of these genes are identical in both the aligned chromosomes. And since a high percentage of DNA sequences are very identical, they are called as homologous pair of chromosomes or homologous pair of DNA. The idea is that at least 100 base pair long DNA should be sequentially similar or identical. So this is a step one where you have two homologous pairs of DNA coming together and aligning with each other such that each gene is lined up one against the other. Then we go to step two. In step two, what is clearly observed is that in the homologous pair of DNA that is present, you would have introduction of a single stranded break, or it could also be a double stranded break. So you may have a cut over here, or you may have a break over here. Both can be cut at the same position. Same goes for here. So either it is a single strand break. In this figure, what you see is a single strand break that is found in both the chromosomes. Now, interestingly, what you would observe is that you have allele B and allele B, and you have small allele B, small allele B. Now, when you look very closely at the sequence of DNA, you may find certain differences. So, for example, here you have a T and an A, and you have a T and an A, but with the small allele where you find a difference is here you have a C and G. So there is a difference in one base pair, but yet majority of the bases are, or majority of the base pairs are identical, and hence these are identical to each other. So some percentage of difference can be possible in the sequence, but when you have the genes being identical more or less, then you would have a homologous pair. So basically step 2 is introduction of a cut or a break in the DNA, which could be either single stranded or it is double stranded. So like mentioned, breakage and joining is an inherent part of the homologous recombination. So here what you can see happening in the step 2 is that there is a break in the DNA. Now step 3 is when from the point where the break was, okay, you have this strand complementarily binding to this strand. So this is important. And why is it important? Because we know that when you have this strand, it is complementary to this strand. When you have this strand, it is complementary to this strand. So therefore, what is very understandable is that this strand will complementarily bind to this strand, and this strand can complementarily bind to this strand. So you can have, therefore, this stretch of DNA interacting with this DNA, while you can have this DNA interacting with this DNA. So there is going to be complementary hydrogen bonding formed over here and complementary hydrogen bonding formed over here. And while they are getting exchanged, this point that is formed is what is called as Holliday Junction or the branch point. So in fact, the step 3 is nothing but one strand of DNA invading the other homologous DNA. So this strand of DNA invades this homologous DNA, and this strand of DNA invades this homologous duplex. So one strand of a duplex is invading the other duplex, and one strand of the other duplex is invading the other duplex. So you basically have one strand each of the duplex invading the other duplex, and therefore in the process of invading the other process you have what is called as a typical cross structure formed, which is called as the Holliday junction. So from this neck that was formed, the strands are able to invade the other pair of homologous chromosome. This is also called as the crossover point, and in certain cases it is also called as chiasma, especially when you refer it to meiosis. So we know that when you have a homologous pair of chromosomes and there are chiasma formed, then you can see how there is exchange of genes. So you can see the red and the white get intermingled or get rearranged because of the formation of the chiasma. So you can see how important or what is the consequence of crossover. Where you had a homozygous or where you had true to type chromosomes, now you have hybrid. So this is something that is very, very evident, and the hybrid is formed only because of this crossover. Step 4. So the branch that was formed over here where there was a cut or where there was a break, that branch will gradually move. And as the branch moves, you can see that more and the more of DNA are getting exchanged between the two. Now, this one can observe it. So when one twists the strands a little, what one can get is a cross. So you can see how this is crossing with this. So you can see how this is crossing with this. Exactly what we are trying to look at, and therefore this is what is called as the crossover or the Holliday junction. Now, this crossover has to be resolved. So step 5 is basically resolution of the crossover. So you can see how this is the crossover. Now this crossover can be cut in two ways. Either it can be cut this way or it can be cut this way. Now when it is cut this way, okay, you would have these two C's coming together. Okay, so these two C's will remain together, but you must understand that this complete C is actually attached to this gray strand. So when you have this being cut, what you will find is that the resultant DNA will be this part. Okay, this will be one pair and this will be the other in the pair. Very effectively, therefore, what you clearly see that the C allele has been exchanged. And between the two DNA, when the C allele has been completely exchanged. Now here you can see that the B allele is not completely exchanged. You only have a small b from this going over here and you still have the capital B over here. So here what you get is heterozygote, but here what you get is a complete exchange of the small recessive C allele to this DNA and the capital C to this DNA. So when you have an exchange like this, then this is what is forming the crossover products. While if the cut is this way, okay, then you do not get a crossover product. You see, you only have an exchange such that the B allele has the small b present over here and the capital B is present over here. So only the B gene is now heterozygous rather than homozygous, while the other two genes remain as it is earlier. So there is no complete exchange of an allelic pair. It is only a partial exchange over here and therefore this is called as a patch or a non-crossover product. So with step 5, when resolution happens, only then, depending on the way it is cut, you will come to know whether you have a complete recombinant or you do not have a complete recombinant. So this is how the Holliday model suggests that where you had breakage, then you also have joining happening, and because you have the joining happening, you have the crossover products being formed. So let us make the conclusions. Gene exchange between two homologous pair of DNA is a natural phenomenon which has been explained by different models, with Holliday Junction being just one of them. The recombination can occur when there is a nick in one of the DNA strands or both the DNA strands. Strand invasion enables formation of the Holliday junction, which looks like a crossroad or a branch point. The branch migration leads to exchange of genes or DNA as the strands from one DNA complementarily binds to the DNA of another homologous duplex. The resolution of the branch point leads to either crossover products or non-crossover products. So Holliday Junction is a road to genetic variation, DNA damage repair, integration of DNA, and is a crossroad that can be resolved to get a recombinant DNA. So can we say that all DNA are recombinant DNA? We leave you with this thought. Thank you.